Hi Hires, we are on the DNA and Genome unit and we're looking at evolution and selection. If you are not totally up to speed with the NAT5, it is worth going back and having a quick recap on the natural selection and speciation from that one. So let's start with what evolution actually is. Evolution is the change that we see in a population's genome over several generations. So it's not a straightforward one generation, oh that's changed, that's evolution. What we're looking for is over many generations. Now for some organisms this might be really fast relative to us. If we're looking at something like a bacteria which has a 20-minute generation time, their evolution might be quite quick in real time compared to ours. Okay. The variation that we're talking about here is the allele frequency, and we're going to use that kind of term every now and again to make sure we're happy with what that means. So what we're talking about here is at a particular gene, whether we have a particular allele that is increasing in proportion in the population or decreasing, and this would be evolution. Now, if this happens over a really long time and we've got a change which has moved it sufficiently far from the original genome, then potentially we get a new species, and this would be speciation. So just to remind you what a species is, it's a group of individuals that can interbreed to produce fertile offspring, and they don't normally breed with other groups. That's important as well. So single generation, you might get an F1 hybrid. They might be closely enough related that you can get a first generation, there we go, we have a combination. But then that next generation doesn't happen. The F1 is not going to be able to breed to produce an F2. And this is all due to the fact that you now have pairs of chromosomes that don't match up correctly when you want to split them to make a half. Zorses can't make more Zorses. So when we talk about inheriting genetic material, the most common thing we say is it's passed down from parents to offspring to next offspring and so on. Now that's actually got a particular name, that is called vertical transmission, because what you've got is DNA that's being passed down through generations. But you also get horizontal transmission, and horizontal transmission is where you can actually pass DNA within a generation, so between siblings of that particular generation. Vertical transmission is what you would normally think of. Parent to offspring, sexual reproduction in eukaryotes, we use that. So what we have is a gamete that has got half the DNA, which then matches up with another gamete with half the DNA required to produce an offspring. And you get a huge amount of variation, and that is really, really useful from an evolutionary point of view. Okay. In asexual reproduction in eukaryotes and prokaryotes, because some eukaryotes like fungi and stuff like that can use asexual reproduction, what they're doing is just passing all of their genetic information from the parent to the offspring. So you're getting a clone, although you may have new alleles being brought in by mutation. So horizontal transmission is mainly a prokaryotes thing, and that's really all you're going to look at in this one. You can acquire DNA in lots of different ways within their lifespan. So that means in one generation, their DNA can change within that generation, and then it could be passed on by vertical, or indeed it could even be moved again in horizontal. Because what you have is the potential by some prokaryotes, they can lift up just bits of DNA that have been released into the environment near them and take them up and then use them. Okay, you can have transfer using a viral system, and this happens quite often. And then we also have one that took a while for us to figure out and know what was going on, where you have DNA that can be moved through a particular plasmid called the fertility plasmid across into the recipient cell. And all of this means that they have the potential for even faster change, even not just to look at the fact that they have a very short generation time, but they're also potentially using horizontal transmission within that generation time. So you've got a huge potential for change. This horizontal transfer means that we have a much, much greater variation within the population, and it's potentially created things like our multi-resistant bacteria. Now, a huge threat to us in terms of how we manage disease, because we've got multi-resistant antibiotics that we can't use anymore because they are no longer being effective against bacteria. And then the short generation time we just get a huge potential for change in a short space of real time. For change to actually happen though, just to remind you, you need selection. So selection, and this is a step up from what you had at the NAT5, it's where the frequency of the alleles increases or decreases in a non-random way. So what you've done is you have pushed a particular allele, either more of it or you've pushed it out of the population. And selection means that you need to then have a particular push, a driver for that. So it has to either be for or against. It could be against a particular allele, but that will allow you to have change. All of this, really, we're talking about Darwin. Wallace was also a big name in this one, coming up with the theory which we can now use to apply to all of this in terms of natural selection. So a basic recap from NAT5. We start off with more offspring than the environment can support. Now that's essential. If you don't have that, then you're not going to have competition because you've not got a limited issue with a resource. Okay, you then have some form of selective pressure. So that's competition of some form that will kill or remove some individuals. The strongest, best suited, fittest, however you wish to see it, to that particular set of circumstances that you now have, survive. And that means they breed, and then we go back up to the top again. We have more offspring than the environment can support. We then have the pressure, the best suited survive, they breed, pass on their alleles. Now what's happening there is a gradual change, and that's what's really important. We've got a gradual change in the population because you get more and more of them with the alleles that were useful to make them survive. Okay, so the allele frequency has changed, and that is evolution. What's really, really important, and you have to stress when talking about this, is that natural selection is non-random, because you can get some random changes to allele frequency, but the natural selection, any form of selection, is non-random. We obviously have as well artificial selection, which is what we do if we're doing selective breeding or similar. That's also non-random. So what we have is a change or a directional change which is going to be reinforced. Measuring evolution is quite difficult, but we can do it if we have a quantitative trait. So a quantitative trait is something which has a particular number associated with it, something you can measure. So things like height and weight and density of colouring, things like that. And if they fall over a range of values, what we know generally from, again, NAT5, is that these are going to be polygenic. They're going to have several genes that are going to be involved in controlling that. It also means that we can see these on a curve, and on that curve we can look to see if the curve moves, and that tells you about what's going on with the genetics. The basic shape of a curve is going to be a bell-shaped curve, and we see this in a lot of different things, but we have what's called normal distribution here. So we have the characteristic going along the bottom, we have the number of individuals going up the side, we have the mean in the center of the curve in this case, because it's completely symmetrical, and then we're moving away from the mean to either side. And overall we have a range of the characteristic running from the top to the bottom. Our selection can now do several things to this curve, and this is the next kind of tricky bit. First one that you have to be able to recognize is something called stabilizing selection. So if we take our original graph, we've got the mean and we have our value of characteristic, and what we have now is a selection pressure that is going to favor the ones in the middle. So what we need is something that too small or too big, or whatever that value might be. It might be height, it could be weight, it could be something with color, it could be something that is not good if you are at either end of the spectrum. So selecting against those two ends means that you actually produce a squeeze on this curve, and the curve changes to a narrowed curve, but the mean hasn't really changed because all you've done is squeeze it from either side. So the average that you take is going to be still where it was. Okay, but what you've got rid of is the upper and lower ends. So if you consider an example of that, that might be birth mass. So in any mammals, actually, you're going to have a much lower mass is going to be more susceptible to other dangers, and a high mass is going to be dangerous for the actual birth itself. So this form of selection you need to recognize. Next form of selection is called directional selection. So here again we have our basic. Okay, and what we mean by directional is it's going to push it in one direction or another. So we are selecting for either this top end is better, or we select for going down the way, and this whole curve will move. Okay, so in this case, if we are selecting for the top end, then the whole curve has shifted up the way and we've now got a bigger numerical mean. Okay, so as an example, we have for this one, European black bears, they increased in mass during each ice age because the larger bodies would have a smaller surface area to volume ratio, which means they lose heat less. So you can see a progressive shift to that new mean. And then when the conditions change, now it becomes more expensive in terms of cost to have a larger body mass, so that mean will shift back down again. Okay, this is not something which doesn't move back and forward according to changes that are happening, and this is really important now when we consider climate change and impacts that way. Your last form of selection is something called disruptive selection. So back to our graph. Now this time, so the first one we had was stabilizing, where it was for the midpoint, the mean. The second one was if we select for one end, either up or down. But then we have this other possibility where instead of selecting for the end, just one of, you select for both options, the top end and the bottom end. Now this has the real potential for creating two peaks. Now if you do that, we've now got two kind of separate points that you might say that we're getting separate means. If this eventually, in the center here, drops down to the point where we've got a gap that doesn't exist in terms of the value, then these alleles in this point here have been completely removed from the population. And that means this population is really distinctly changing. And once we have that, potentially you could go to speciation. Okay, and this is important, and we'll look at when we look at speciation forms in later bits, in particular sympatric ones.